All right, let's talk through this last part of chapter 11, starting with operon. So this is where you really want to pay attention. That's why I stopped the video and started a new one, so that this was at the beginning of the video. So this is super important. So operons are found only in bacteria and archaea. So it's a coordinated set of genes regulated as a single unit. It can be induced or repressed. So we're going to talk about both instances. Categories determined by how transcription is affected by the environment surrounding the cell. So things that are on the outside of the cell will either bind or induce this operon and make it either start or shut down. So an inducible operon is catabolic. So that means it's going to break things down. It's going to, the operon has enzymes for catabolism. So it's going to break things down. So the operon is turned on or induced by the substrate that needs to be broken down. Enzymes needed to metabolize a nutrient are only produced when the nutrient is present. So induced is catabolic. Repressible is when you need to make things. So it's anabolic. So several genes in a series are turned off or repressed by the product. So if it's already there, you don't need to turn it on. So the ones that we're going to talk about, this one's the lactoperon or the lactoseoperon. It has a regulator that's composed of the gene that encodes for the repressor, which is a protein capable of repressing the operon. You have a control locus, which has a promoter, which recognizes the RNA polymerase, and an operator that acts as an on-off switch for transcription. There's also a structural locus made up of 3 genes coding for different enzymes needed to catabolize lactose. So we're going to break it down. Okay. So the repressor protein is allosteric. So you have 2 binding sites, one for the operator sequence on the DNA and another for lactose. In the absence of lactose, the repressor binds to the operator blocking transcription for the structural genes. The regulator gene lies upstream of the operator and is transcribed constitutively. So it looks something like this. So you have a regulator that is going to affect the repressor. So you have a promoter and an operator. So it is going to, if the repressor is there, it's going to stop transcription. Okay, so it's normally off. That is what it is. So you don't need those genes to make the to break down lactose. But if lactose is there, it's going to induce it and it's going to bind to the repressor and make it fall off. And that's going to allow the RNA polymerase to bind to the operator and then make those genes. As long as lactose is present, the repressor is going to be off and it's going to be able to make the enzymes to break down lactose. If lactose is depleted, the repressor is going to be able to attach and it's going to be shut down because you don't need those enzymes if lactose isn't there. All right, so the difference between an inducible and a repressible operon is repressible operons are usually in the on mode. So they'll only be turned off when the nutrient is no longer required. Excess nutrients act as a co-repressor to block the action of the operon. So how this one works, this is going to be the arginine. It can also be the tryptophan operon. So that's a lot of the times what you'll see is either tryptophan or arginine in bacterial cells. So how this one works is it's going to go ahead and make those genes to make arginine, and the repressor is going to be inactive. So it's just on, okay? But when arginine accumulates, it's going to bind to the repressor and then it's going to attach to the operator and it's going to shut it down. So once the arginine is depleted, then it's going to go back to its inactive state for the repressor and then it'll fall off. I have several videos here and those are all posted in the announcement or in the content guidance. So phase variation is a result of bacteria turning on and off a complement of genes that leads to phenotypic changes. So they can actually change what they look like in order to, evade the immune system or make them more virulent. So they're heritable, they're passed down to subsequent generation, and it involves the turning on of genes mediated by regulatory proteins as described with operon. So very similar. Antibiotics can affect transcription and translation. So drugs can inhibit protein synthesis, like rifamycins, actinomycin D. There's also ones that interfere with the ribosome, like erythromycin, spectinomycin, chloramphenicol, lincomycin, tetracycline, and aminoglycosides. Then we're going to get into recombination events and horizontal gene transfer. So recombination is an event in which one bacteria donates DNA to another. The end result is a strain different from both the donor and the recipient. Plasmids are normally what are being moved between them, and though they are extra chromosomal DNA, adept at moving between cells. So a recombinant is any organism that contains and expresses genes that originated in another organism. Horizontal gene transfer, we're going to talk about several different ways that occurs. So it's DNA that is transferred between organisms, and new genes are acquired that don't come from the parent. So it's like siblings or the same generation transferring genes. So plasmids are small circular pieces of DNA. We will talk at length about plasmids in chapter 12 as well. That can replicate independently of the bacterial chromosome. They allow the transfer of DNA between cells, found in most bacteria and in some fungi, contain at the most, a few dozen genes. Not necessary for survival, but does carry virulence factors. So if you're doing your SLP right now, you're probably talking about virulence factors, and they're going to be on those plasmids most likely. There is a video that's linked in your announcement or your guidance on horizontal gene transfer. So we have three main types. Conjugation, which is going to be in the form of a pilus, that's going to be direct. So it's like an arm that sticks out and the plasmid goes between. It's like a tunnel. And some examples of genes that can be transferred are drug resistance, resistance to metals, toxin production, enzymes, adherence molecules, degradation of toxic substances, and uptake of iron. Transformation, we talked about this with Griffith's experiment, if you remember back to that. It's when free donor DNA fragments are brought into the cell. They just are absorbed through the membrane, and they're live, competent recipient cells. So they're indirect. They can be polysaccharide capsules and unlimited with cloning techniques, and we'll talk about that next week or next chapter. Transduction is when the donor is a lysed bacterial cell and a defective bacteriophage is the carrier of the donor DNA, or there's live recipient cell of the same species as a donor. So a lot of the times this is through a phage. It's indirect, and it can be toxins, enzymes for sugar fermentation, and drug resistance. So let's go through them. So conjugation is like bacterial sex. It is a mode of genetic exchange with a plasmid or other genetic material that is transferred through a pilus. And it is through direct connection, so a tunnel. Can occur in both gram-positive and gram-negative cells. So this is what this looks like. So we have this F factor transfer. So the F factor is replicated and a bridge is made between the F positive and the F minus, and it gives a copy of the plasmid to it through this tunnel or through this pilus. A high frequency transfer is when chromosomal genes are transferred. So this is a plasmid. The F factor is a plasmid. This is actually chromosomal genes that are transferred. It can be partial or it can be the whole chromosome. Okay. Most of the time it's just parts of it because they can't handle a huge chunk. And then it incorporates into the other chromosome. Conjugation has resistance plasmids. So these carry genes for resistance to antibiotics and other drugs. Commonly shared among bacteria through conjugation, can confer multiple resistance to antibiotics. Our resistance factors can also carry genetic codes for resistance to heavy metals or for synthesizing other virulence factors. And here's some videos on conjugation that you can watch. Conjugation is also can talk about the genome and how that's mapped. So how that works is they time it. So when you watch that video, it actually times it so you can see when genes are transferred through. And that's how they map the genome of the bacteria. Transformation is capturing DNA from a solution. So transformation is the acceptance of a bacterial cell with the fragments of soluble DNA that's just floating around. So basically a bacteria dies, or you just put donor DNA into the solution, and then it just accepts it through the membrane. They're normally called competent cells, so they're capable of accepting genetic material. Sometimes you have to electric shock them to get them to open up to give that. Sometimes you can heat shock them too, make the cells really hot, and then they open up and allow that DNA in. Discovery of transformation. We talked about this before with Griffith's experiment. He had encapsulated and non-encapsulated strep pneumoniae in laboratory mice. So he had encapsulated strains have the smooth colony, and the strains that lacked the capsule were called rough. And he mixed them together. So he had these live strains would kill the bacteria, the capsule strains, but the non-encapsulated would not kill the mice. So he heat killed the ones with the capsule and mixed them with the live strain, and the DNA was given to the rough, and it killed the mice, and they were able to um, gather up the strains that were both smooth and rough from the mice that died. There's some videos on transformation. Transduction is used with a bacteriophage that serves as a carrier for the donor recipient. Donor cell to the recipient naturally occurs in a broad spectrum of bacteria, because remember, a bacteriophage is a bacterial virus. The participating bacteria in a single transduction event must be the same species. So you can have generalized, which is random fragments. It's not directed. So when we say generalized, that's random. So random fragments of disintegrated host DNA are taken up by the phage during assembly, and any gene from the bacteria can be transmitted. So basically, the phage lands, injects, and the DNA will then make new phage, and then they're lysed, and it keeps going on and on and on and on. Specialized is when a highly specific part of the host genome is regularly incorporated into the virus. So it's like specific genes are inserted. Explained by the prior existence of a temperate phage inserted in a fixed site of the bacterial chromosome. So a prophage DNA separates from the chromosome and carries the genes with it. So here you've got the prophage, and then it is going to be excised from the chromosome and then make phage, and it's going to break open and then go to the next cell. There's some videos here on generalized and specialized transduction. Another way is transposable elements or jumping genes. We call these TEs or transposons. They're jumping genes that shift from one part of the genome to another. It was first proposed by Barbara McClintock in 1951. Jumping genes are widespread among cells and in viruses. So these genes just pop out, and then they're able to be transferred within cells. Types of transposable elements, you've got insertion sequences. So these are the smallest TEs, consist only of 2 tandem repeats. You can have a retrotransposon that's going to be a type of TE that can transcribe DNA into RNA and then back into DNA for insertion into a new location. Other transposable elements can contain genes that code for antibiotic resistance and also toxin production. Effects, so it scrambles the genetic language, can be beneficial or adverse depending on where it inserts, what kind of genes there are, and what type of cells are involved. Effects in bacteria, changes in colony morphology, pigmentation, antigenic characteristics, replacement of damaged DNA, and transfer of drug resistance between bacteria. So there's some videos here that you can watch for those. There's also a pathogenicity island that can make the ability for the host to transfer multiple genes at once. So they're coordinated, they're lined up, and so the islands are flanked by sequences that look a lot like genes for transposable elements. And so they're able to take out that whole group of them and move them to different bacteria. And then the last part is mutation. So this last part, we're going to talk about changes in the genetic code. So mutation is any change to the nucleotide sequence in the genome. It's the driving force for evolution. In microorganisms, it becomes evident in altered gene expressions such as altered pigment production or development of resistance to a drug. Mutations, there's a wild type, which is the normal, natural, non-mutated strain. And then the mutant has the mutation. So the wild type is going to be the trait that's present in the highest number in the population. So it can be spontaneous or induced. So spontaneous is just a random change in the DNA from errors in replication and occurs randomly. Induced is from a mutagen. So some chemical mutagens are nitrous acid or bisulfate, which removes the amino group from some of the bases. Ethidium bromide, it inserts itself between the base pairs. And they actually use ethidium bromide a lot in labs to make it so you can see the DNA very well. Acridine dyes cause frame shift due to insertions between base pairs. Nitrogen base analogs compete with the natural bases for those different sites on replicating DNA. And then we have radiation. So you have ionizing like gamma rays and X-rays that form free radicals that cause single or double breaks in the DNA. And then UV, which we actually do in a lab that we did very recently, and it causes these cross-linkings between the pyrimidines or the thymine dimers. So it causes these cross-links. And then there's some videos on mutagens, different types of categories of mutations. There can be point mutations, which are small mutations that affect only a single base on a gene, involve the addition, deletion, or substitution of a single base. They can be lethal or neutral. So lethal would lead to cell dysfunction or death. Neutral, it's neither adverse or helpful. So it doesn't have really an effect. More categories, there's a missense, which any change in the code that leads to the placement of a different amino acid. So if it's like a glycine and then it's an alanine, that would be a missense. Nonsense changes a normal codon into a stop codon, so it makes it shorter. Silent mutation alters a base but does not change the amino acid. That's from that redundancy of the code. That does not alter the amino acid sequence. And then a back mutation occurs when a gene has undergone a mutation and then it reverses back to the original base. Frameshift mutations occur when one or more bases are inserted or deleted from the DNA strand. This alters the reading frame of the mRNA. Nearly always results in a non-functional protein. Every amino acid after the mutation is different from what is coded in the original DNA. Insertion of bases in multiples of 3 would not disturb the reading frame, but would change the protein. So there's some videos here you can watch on mutations. Then there's also repair. So in the UV radiation lab that we did, we wrapped our plates in aluminum foil, and that's to make it so we can't have back mutations or repair. So there's these things called photo activation or light repair that will reverse the damage caused by UV radiation. There's also excision repair, which is where enzymes come in and correct the bases and add in the correct ones. Mismatch repair is a repair system that can locate mismatched bases that were missed during proofreading, and then the base must be replaced soon after the mismatch is made, or it will not be recognized by the repair enzymes. Again, there's some great videos. The Ames test, which I love this. This is a test question, just so you know, because I'm from Ames, Iowa, and it just reminds me of that. So commonly used to detect chemicals that are carcinogenic. It uses bacteria rather than experimental animals, allows easy observation and monitoring of gene expression and mutation rate. Any chemical capable of mutating bacteria could similarly mutate mammalian DNA and is potentially hazardous. So what we have here is a salmonella bacteria in a histidine negative media. Because the control setup has the bacteria plated on a histidine-free media containing liver enzymes but lacking the test agent. So your control plate, it's going to lack histidine and the test chemical, and these histidine positives are arising from spontaneous back mutations. The experimental plate is the same, but it contains the test agent. So it's a minimal media, and then that has no histidine, incubates it with to form that back mutation, and then if it has this degree of mutation, mutagenicity, oh gosh, I can't say that, can be calculated by comparing the number of colonies growing on the control plate versus the test plate. So chemicals that induce an increased incidence of back mutation are considered carcinogenic. So whatever this was on this test is drastically larger than the control. So there's positive and negative effects to mutations. So mutations that are not repaired can have effects on the nature of, it depends on the nature of the mutation and the strategies available to the organism. Mutations are permanent and heritable. They're passed on to the offspring or new organisms or new viruses, and they enter the gene pool and become a long-term part of that. A small number of mutations contribute to the success. So that's like the survival of the fittest. A variant strain with an alternate pathway of expressing a trait can be more readily adaptable and survive and reproduce. So the whole goal is to get to reproductive age. And that's it for chapter 11.